Hello everyone. Let us proceed further in transcription mechanism. So in the last lecture I talked about the overview of transcription process. In that an RNA polymerase would bind to a promoter region of the gene to be transcribed, and it opens up the DNA strands forming a transcription bubble and thus initiate transcription process. Then it slides through the gene and carry out elongation of RNA molecule. Then at the termination site, RNA polymerase leave the RNA strand with simultaneous release of the RNA molecule. Now this RNA polymerase is again used for another round of transcription process. So see, during transcription, many RNA copies can be made from a same gene in a relatively short time. So it is not the case that one gene would transcribe only one RNA molecule. Rather, one gene can form number of RNA copies, and that too in a relatively short time. That means it is not that RNA polymerase would bind the promoter region, carry out transcription process, and when the transcription of one RNA molecule is complete, it would leave the termination site and then again bind the promoter region and then again carry out the transcription of one RNA molecule. No, rather the uh, the transcription process happens. The number of RNA copies are made in relatively short time. Many copies of RNA molecules, they are made in relatively short time because many number of RNA polymerase would simultaneously transcribe a gene. Okay, so let us understand this better by looking at this figure. So this is a micrograph showing the simultaneous transcription of two adjacent genes. So this is the transcription of two adjacent genes as observed under the electron microscope. So it is a micrograph where you can see the transcription process happening in two adjacent genes. So here you can see that this is one gene and this is another gene, and there are dots on the genes, and these dots have fine threads attached to them. Okay, so these dots are RNA polymerase molecules moving on the gene, and these threads are the RNA transcripts. Okay, so you can see that simultaneously number of RNA polymerase molecules are bound to the gene and carrying out the transcription process. Okay, so this picture is of transcription of ribosomal RNA molecules which are not translated further, and these are used for ribosomes. These are components of ribosomes and which carry out or which facilitate translation process. Okay, and another point is that in both the genes the transcription process is happening from left to right. Okay, transcription is happening from left to right. As you can see from in the left side, the transcripts, the RNA molecules are smaller in size, and as you go further towards the right, you see the longer RNA threads, the longer transcripts, and at the end of the gene, you see the longest RNA transcripts. Okay, so this is it. This is a very beautiful picture about the transcription process happening, and this gives a very clear idea that how transcription process goes on within the cells. So this picture I've taken from Albert 6th edition, figure 610, page number 305. So each RNA polymerase molecule would move at a speed of 50 nucleotides per second. Okay, so I hope this is clear. Moving further. So another point to be discussed is that RNA polymerase lack proofreading. So RNA polymerase lack a separate proofreading 3' to 5' exonuclease active site as in DNA polymerase. So DNA polymerase has this 3' to 5' exonuclease active site, but RNA polymerase lack this separate proofreading active site. So the error rate of transcription is higher than replication. So the RNA polymerase make about one mistake in every 10 raised to power 4 nucleotides copied into RNA, compared with the error rate for direct copying by DNA polymerase of about 1 in 10 raised to power 7 nucleotides. So in transcription, one mistake is made for every 10 raised to power 4 nucleotides. In replication, one mistake is made by DNA polymerase in 10 raised to power 7 nucleotides. So in case of the mistake, all RNA copies are degraded or replaced. But still, the RNA polymerases, they have a modest proofreading mechanism. A modest proofreading mechanism is there in RNA polymerases. So what is that? It is that in both bacterial and eukaryotic RNA polymerases, they do pause when a mispaired base is added during transcription. And they can remove the mismatched nucleotides from the 3' end of a transcript by direct reversal of the polymerase reaction. But it is not yet clear that this activity is the true proofreading function. Now let us have a look on the structure of the RNA polymerase of E. coli. So this is DNA dependent RNA polymerase, and so this is a large complex enzyme consists of 5 core subunits. So 5 core subunits, that is alpha 2, beta, beta prime, omega. So 2 alpha subunits, 1 beta subunit, 1 beta prime, and 1 omega subunit. The molecular weight of these subunits in total is 3,90,000 Daltons. And there is a 6th subunit which is variable in size, and that is sigma subunit. So there are a number of sigma subunits in E. coli. The most common subunit is sigma 70. The molecular weight of sigma 70 is 70,000. So sigma 70, the 70 represents the molecular weight of the sigma subunit. So let us have a look on different sigma subunits of E. coli. But before doing that, let us have a look on the function of sigma subunit. So sigma subunit binds transiently to the core and directs the enzyme to specific binding site on the DNA. So sigma subunit is important for binding of the RNA polymerase on the promoter regions of the gene to be transcribed. So sigma subunit is important for tight binding of the RNA polymerase molecule to the promoters on the DNA molecule. So this is required for binding and the tight binding. Without sigma subunit, RNA polymerase would loosely bind the DNA molecule and won't be able to carry out the transcription process. So this we'll discuss this in more detail, and let's have a look on the. So this table shows the 7 sigma subunits of E. coli. So E. coli has 7 different sigma subunits. So these are different sigma subunits: sigma 70, sigma 54, sigma 38, 32, 28, 24, 18. These are the molecular weights of the sigma subunit. And here is the frequency of these molecules present in the cells. So sigma 70 is present in a very high number, around 700 molecules in a cell at a time. And the function of sigma 70 is housekeeping, and like that, uh, different sigma subunits have different functions. Sigma 54 has modulation of cellular nitrogen levels. Sigma 38 is used for stationary phase genes. Then sigma 32 used for heat shock genes. Then sigma 28 is for flagella and chemotaxis genes. Then these are present in less frequency, and they have sigma 24 and sigma 18. They are used for extra cytoplasmic functions, some heat shock functions, some extra cytoplasmic functions, and other. Like different sigma subunits, they have different kinds of functions. Now, what do we mean by function? That means that these subunits would bind to the promoter regions of the genes involved in these kinds of functions. Okay, so sigma 70 would bind to the promoter region of the gene involved in housekeeping functions. Likewise, sigma 32 would bind to the what would help in binding of RNA polymerase to the gene which are expressed, the heat shock genes, which are expressed in the heat stress. Okay, so suppose if there is a heat shock, when there is a heat shock, the cell, these genes would be expressed. Okay, heat shock genes would be expressed, and uh, these genes will not be able to express if sigma 32 is not bound to the enzyme RNA polymerase. So the core subunit is same of the core subunit of RNA polymerase is same, that is alpha 2, beta, beta prime, omega, but the sigma subunit is different. Okay, there are different sigma subunits for different kinds of genes. So let us go back. So these 6 subunits, the 5 core subunits and 1 sigma subunit, constitute the RNA polymerase hollow enzyme. So it is the RNA polymerase hollow enzyme having the 6 subunits that would bind to the DNA molecule for transcription process. Okay, so let's go further. Now let us have a look on the structure of the RNA polymerase enzyme. So this is the structure of RNA polymerase hollow enzyme of the bacterium Thermus aquaticus, which is very similar to that of E. coli RNA polymerase. So you can see different subunits in different colors. So the beta subunit is beta subunit, which is in gray color. Then there is beta prime subunit, which is light gray color or white color. And there is this 2 alpha subunits showing showing in different shades of red. Then there is sigma subunit, which is transiently bound to the other subunits, the core enzyme. This is in brown color, and omega subunit is the smallest subunit, which is seen when you when this enzyme is turned 180 degrees. Then you can see the omega subunit, which is in the some greenish shade. Okay, so this is about the structure of RNA polymerase hollow enzyme, and let's move further. So RNA synthesis begins at promoters. So the RNA polymerase binds to specific sequences in the DNA called promoters, which direct the transcription of adjacent segment of DNA, that is gene. So binding of RNA polymerase to promoter region of the gene is crucial and critical. So in E. coli, RNA polymerase binding occurs within a region stretching from about 70 base pairs before the transcription start site and about 30 base pairs beyond it. So DNA base pairs before the transcription start site, that is the upstream of upstream DNA molecule. So the DNA base pairs upstream the transcriptions start site are given negative numbers, and DNA base pairs downstream the transcription start site are given positive numbers. Okay, so therefore the RNA polymerase binding occurs within the region stretching from minus 70 base pairs to plus 70 plus 30 base pairs. So the binding region spans from minus 70 base pairs to plus 30 base pairs. So let us have a look at the sequences present at promoters to which the sigma subunit of RNA polymerase hollow enzyme binds. So there are special sequences present at promoters to which sigma subunit of RNA polymerase hollow enzyme binds. So this figure shows typical E. coli from promoters recognized by an RNA polymerase hollow enzyme containing sigma 70 subunit. So the common bacterial promoters recognized by RNA polymerase hollow enzyme containing sigma 70 were analyzed and compared. The analysis and comparison have revealed the similarities in two short sequences centered about positions minus 35 and minus 10. These sequences, the minus 35 region and minus 10 region sequences at minus 35 region and at minus 10 region. These sequences are important interaction sites for sigma 70 subunit of RNA polymerase hollow enzyme. So these regions are also called boxes. This region can be called minus 35 box, and this region can be called minus 10 box. So, however, these sequences are not identical for all bacterial promoters. So bacteria has different genes, number of genes, and all genes have promoter regions. But the sequences, they are not identical for all bacterial promoters. But the nucleotides at the minus 10 and minus 35 regions form a consensus sequence. So the consensus sequence at minus 10 region is TA-T880, that is Tata box, and at minus 35 it is TTGACA. And there is another recognition element apart from these two regions, there is another recognition element called up element. Up means upstream promoter, which is upstream of these regions. This is up element, the upstream promoter, and this is another recognition element, and this is AT rich recognition element. If you look at the sequence, you will find the sequences are AT rich. This region is AT rich, and it occurs between positions -40 and -60. And this UP element is not expressed in all. This is not present in all the genes. This UP element is present in the promoters of certain highly expressed genes. Few genes which are highly expressed in bacteria have the up element, which is a AT-rich recognition element. This up element is bound by the alpha subunit of RNA polymerase. I am repeating that this up element is bound by the alpha subunit of RNA polymerase. And you can see there is a spacer region between the -35 region and -10 region, and -10 region in the RNA transcription start site. So these spacer region, they vary in number of, there is a slight variation in number of nucleotides, as the number of nucleotides in this spacer region between -35 and -10 is from N16 to N18, and the spacer region between -10 and the transcription start site, this is from N6 to N8. So the spacer arm between -10 region and RNA start site has 6 to 8 nucleotides. So this is it for today, and we will discuss more details about transcription mechanisms in the next lecture, a few more lectures. So goodbye.